Thank you for the introduction, and thank you all for coming. Now, for this project, I use real COVID-19 data, which is from January 22nd, 2020 until a couple of days ago, actually, February 11, 2021. This data is provided by the Johns Hopkins University Center for System Science and Engineering. But first, we do some exploratory data analysis, although the main goal is really to use the data to fit the SIRD model parameters using least squares, and then visualize and compare the solution to the fitted model with the actual data. In this talk, however, I'll focus on the computational side of the project and the technology that I'm using, and that's the R programming environment with R RStudio as the IDE, the integrated development environment. That's just the editor. It's a really good editor. And I use R Markdown notebooks basically to kind of as the main documents that contain the project narrative, mathematical LaTeX expressions you can put in there, code. And then when you knit this document into a PDF file, you kind of generate almost publication quality document where everything is knitted and sort of seamlessly embedded into the final document. You've got the code, the numerical, graphical results, everything is embedded there. And this is what I used in the upper level courses that I teach. It takes me usually a lecture to show the students how to use this technology, and they use it to generate all their reports for homework, projects, exams, everything. I use it for my lectures to develop, and it's sort of a one-stop solution for me. In fact, even this presentation I created with this technology. Now, a little bit more about the data. Now, the Johns Hopkins University Center for System Science and Engineering created a publicly available GitHub data repository. So they're famous with their dashboards that probably everyone has seen by now. And they consolidate data from various sources, national and international. However, instead of downloading sort of data files from their GitHub repository, I'm using the coronavirus R package because somebody already sort of created an R package that pulls the raw data from the GitHub data repository of Johns Hopkins University. And the name of this package is coronavirus, which kind of has all the data that I'm using. Now, it's very easy to use the data. All you have to do is basically install the coronavirus package in RStudio and then just load the data. And all this you can accomplish with these 2 lines of code that you can embed inside the Markdown document and run. Assuming that you've already installed the coronavirus package, then you load the library, and then with the data, the name of the data set is also coronavirus, you make the data available for you to start playing with. So it's pretty convenient that way to kind of use any data sets. And since there are a lot of R packages that provide a lot of data, but even if there's no R package, it's also very easy to sort of download and install data from a website with just 1 line of code. You can download and load the data and start using it basically. And just to kind of give you a little bit of taste of that, we'll do some exploratory data analysis for us. And so once the data is available in RStudio, you can kind of pick into the data by using the head function. So this entire table was created just by typing head inside coronavirus, which by default shows the first 6 rows of the data. And you see the variables: date, province, or state, country, latitude, longitude, type, and cases. So the data actually has 7 variables with more than 300,000 observations between January of last year and February now. And I'm using for this project the date variable, the country variable, the type, and the cases. The type variable in particular has 3 possible values: confirmed cases, so infected, recovered, and death. Now, for the data analysis, I use the dplyr package, which is part of the tidyverse collection of R packages that was actually developed by RStudio. This is a very modern, very popular collection of packages for doing modern data analysis and visualizations. Now, once you install and it's a 1-click install to install the entire tidyverse collection inside RStudio, and then you load the library. And then you can sort of start working directly on the data. And I want to give you a little bit of a taste of what it takes to do sort of data analysis of such big data. and it's I'll show you several tables. This is the first of those tables that sort of displays here the top 8 countries in terms of the total confirmed cases as of February 11, the last time I updated this. And you can see, unfortunately, US is on the top with more than 27 million total confirmed cases, then India with close to 11 million, Brazil close to 10 million, UK, Russia, France, Spain, Italy, and so on. So this entire table, it was created with this code. Just to kind of give you a taste that it doesn't really take that much to kind of to do the data analysis. So I'll just go very quickly over the code just to again give you a sense of how data analysis is being done. Now, the coronavirus is the name of the data. So this is actually, it's called the pipe operator, and it's kind of a modern invention. It's really the equivalent of function composition. So basically, and you read it like that, from the data, then group the data by country and type, and then summarize, create a new variable called total cases by summing the cases variable, then arrange in decreasing order the total cases. And that's it. And then from this data frame, then filter by type confirm. So extract only the confirmed cases and then show the head of the data with only the first 8 rows, and then create this table. And this is the actual code that created this table. I didn't actually typeset this table manually. So it was all automatically created and embedded into the presentation from this chunk of code. Now, here are several other tables. This one shows the top 8 total death cases as of February 11. Unfortunately, US again is on the top with 475,000. As of today, is actually above 480,000. Then Brazil, Mexico, India, the UK, Italy, France, Russia. Another table that shows the top 8 countries in terms of the recovered cases. India on the top. Then Brazil, US, Russia, and so on. Now, the recovered cases, though, are considered to be sort of systematically underreported. Now, finally, I'll show here the global totals as of February 11. So in the world, and this is the code that produces this, it's very compact code that produces the global totals across all countries and across all times. The number of confirmed is more than 107 million people. The number of death cases, more than 2 million. The recovered, more than 66 million. Now here I created a couple of also interactive plots that is relatively easy to create with the ggplot library, which is part of the tidyverse, and the plotly. This is a plotly plot that was created by converting a ggplot into an interactive plot. So you can basically sort of hover on the top and see interactively information about the data. I was curious why these 2 peaks here in December for both the total cases, about 1.5 million confirmed cases, and 2 days later on December 12, a very similar number of 1.5 for the recovered. But this is sort of the time series for the 3 types of cases: confirmed, death, and recovered. Here I plotted the global cumulative confirmed, recovered, and death cases. The red is the confirmed, so that's the recovered and the death on the bottom, because it's sort of a different scale, really, than the recovered and the confirmed. So you can kind of see the evolution over time for the global cumulative confirmed cases. And now this is just for the US. You can extract the data, you can filter the data only for the US, and this plot shows the cumulative confirmed cases in the US. You see the spring, basically the spring sort of increase, and then the summer increase, and finally the fall increase in terms of the cumulative cases. Now, what I want to do next is basically use this real COVID-19 data in the US to fit an SIRD model, which is sort of a simple generalization of the classical SIR model that was developed in 1927, in fact. And in addition to the susceptible, infected, and recovered populations that are involved in the SIR model, I sort of introduced also the 4th category for the deceased, which is the total number of individuals at time t who have died from the disease. And this is the system of differential equations that defines the model. And again, it's sort of a simple generalization of the SIRD model. where the total population is assumed constant, and the parameters of the model here, we have three parameters, beta, gamma, and mu. The beta, this is the rate of infection, the gamma is the rate of recovery, and mu is the rate of mortality. So I will show you how to basically fit these model parameters for this relatively simple model, which is a simple generalization of the classical SIR model, how to fit the parameters to the real data. And so note first that the variable I in the model as a function of time represents the cumulative number of infected cases at time t, and the variable d represents the cumulative number of death cases. And so, which means that I kind of have to use then to extract from the data the vectors with this the cumulative number of cases of infected and the death cases. But before we do that, so I want to show you how you can find numerical solutions of systems of ODES using the DSolve package in R. Now, for the DSolve solver, basically the key ingredient is to provide the model. And this is the function, this is an R function that I call, the name of the function is SIRD, R function that basically provides sort of the implementation for the model. And you can see here that on the left, you have the differentials for the four key variables SIRD, and on the right-hand side, you have basically the right-hand side of the model. So it's relatively simple to implement the numerical solutions of this system of ODES using the DSolve package. And this is the first ingredient that you need, the function that implements the model. So all you do is sort of put the differentials for the key four variables here and assign the right-hand sides from the model, where you use the same names, beta, gamma, and mu for the parameters. And then in addition to that, and this chunk of code that you see here, it provides, in addition to this previous function, SIRD, this chunk of code provides the full implementation to actually find the numerical solution of any really system of ODES. It's very straightforward. So here I start with, I load the library DSolve, and I start with the initial values for the, I chose 50 million for the size of the full population, initial population being exposed. And then in it is a vector of values for the number of susceptible, which is sort of, I chose to start with 33 values of infected people initially. Then you need a vector of for days. This is sort of a vector of integer values from 1 to 30. Then you need to provide a vector for the parameters. I chose this sort of pretty much random, not quite, they're kind of close to the fitted values though. And then it's very important that you name these parameters to the same names that are used in the model function, beta, gamma, and mu. And this is the key line that provides the numerical solution to the system of ODES using the ODE solver from the DSolve package. So basically you pass in the vector of initial values, then you pass the vector of days. In this case is just from 1 to 30. And then for the function, you pass the SIRD function from the previous slide that sort of implements the model. And then you pass the vector of parameters here that I chose this vector with this, the values of the three parameters. And again, it's very important that the values are named. Beta, gamma, and mu, so that you have consistency with these values here that appear beta, gamma, and mu in the model function. And that's it. So calling this the ODE solver will basically return the solution, which is really this matrix that you see here that I print with this command so that you can see the output. It is just a matrix with 1, 2, 3, 4, 5 columns, starting with the time, and then the S variable I, R, and D. And we're going to use this solver basically to do the optimization using least squares. So we want to really minimize the sum of square differences between the data and the model solution with respect to the model parameters beta, gamma, and mu. But we'll fit the model only to the observed cumulative infected and death cases, not to the recovered cases. And the reason is that the recovered cases have been considered systematically underreported. And so the residual sum then of squared the residual sum of squares, the RSS, as a function of beta, gamma, and mu is then defined this way, basically, where here the infected stands for the vector of cumulative infected cases extracted from the data. So these are the actual observations. The I vector is the vector from the previous, from the matrix here that comes from the solution of the model for the particular set of parameter values. Here, def would be again the vector of from the actual data of cumulative def cases, and the capital D here would be basically from this matrix, the last column of this matrix that comes from the fitted model, from the model for the specific set of parameters. So we're trying to minimize this function basically, where again this part comes from the data, and this part comes from the model for the particular set of parameter values. Here, capital M stands for the number of observations in the training set, in the training data set. I already mentioned that the infected K and death K, these are the K components of the observed cumulative infected and death cases, respectively. The I sub K, D sub K are the K components of the solution vectors, or the third and the fifth columns of the ODE solution that is produced by the model for a given set of parameter values. And this is all the code that is being used to do the model fitting, to implement the model fitting. So for the days, I create a vector sequence along the vector of infected will create basically the integer sequence that would serve for the sequence of the days, and 50 million the initial values. Then the vector of initial values for all the variables S, I, R, and D, and then this is the key function, which is the residual sum of squares that we want to minimize. So this is how you implement. This is the R function that implements basically this function that you see mathematically here given in on the previous slide. And so it's a function of the parameters, and the parameters is assumed to be a vector. But first we name these parameters to beta, gamma, and mu as before, and then we get the solution of the ODE based on the vector of initial values, the vector of days, the model function, and the vector of parameters. And now the I variable is from the solution matrix, the third column, as I mentioned earlier. The third column specifies actually the solution for the number of infected cases. And the D variable would be the fifth column. This represents basically this syntax that represents the fifth column in the solution matrix that comes from the numerical solution of the system of ODEs. And then the key line here, this sum that you see, in code is implemented very easily with this line because it's a vectorized code. So you just take the infected, which is the name for the vector of cumulative infected cases from the observed data. The I is basically the vector of cumulative infected cases from the model. So you take the square. Similarly for death and D, you take the square, and then you take the sum. And this expression in code is a vectorized version of this mathematical expression, and it sort of returns actually this sum. And this is the function that we want to minimize. And we use the base R general purpose optimizer, optim. And let me show you here the code that implements the optimization. So optim is the general purpose optimizer. Then you pass a vector of initial values for the three parameters. Then you pass the name of the function that we want to minimize, the residual sum of squares. And this method here that I've chosen is basically a quasi-Newton method that allows to provide lower and upper bound because these are rates. So you can put them in the interval 0, 1. And that's it. This does the optimization. It minimizes the function. It gives you the optimal values of the parameters, beta, gamma, and mu. Beta is 0.67, gamma 0.32 something, and mu 0.0064. Mu is the mortality rate. Now, a very important sort of parameter that is associated with the model is the basic reproduction number R0, which represents the expected infected cases generated by one infectious case. And in particular, if this value, if the value of R0 is bigger than 1, it kind of predicts an epidemic. Now, R0 in general can be computed by analyzing the threshold between the stable and unstable equilibrium of the model using the eigenvalues of the Jacobian at the free equilibrium. And if you do that, you get this formula for R0, which is beta divided by gamma plus mu. And for the numerical values of the optimal parameters, it's actually very close to 2. I kind of looked a little bit in the literature to see what people have done in research publications. They've actually used, I was surprised that in research publications, people have used the SRRD model to compute the basic reproduction number, and was found to be in the interval between 1 and 2.8 That's based on one study for a number of different countries with mild to severe cases of their local epidemics. That's the paper basically that sort of has this information. At the end, I wanted to show you how well the fitted model actually compares to the observed data. I was actually honestly surprised that the fit was so good. So the fitted model is represented by the curve in red. That's the fitted model, and the blue dots represent the data points. So these are the data points for the observed cumulative infected cases, and the red curve basically represents the cumulative infected cases predicted by the model, by the fitted model. So it's a pretty good fit, surprisingly good fit, in fact, for this sort of simple, relatively speaking, model. And also on this graph, I wanted to show you a similar fit, but for the cumulative death cases. So again, the red curve represents the fitted SIRD model in terms of the cumulative death cases, and the blue dots represent the data points. So this is based on the training data set from the first 3 weeks of March. And it's a pretty good fit. Now, in conclusion, I'll finish with, I think I need a couple of more minutes. Now, in general, for simple models, the fraction of the population that needs to be effectively immunized to prevent the spread of the pandemic, known as the herd immunity threshold, has been estimated. I found it in a paper. I could actually add the reference here to the paper. Has been estimated to be larger than 1 - 1 over the reciprocal of the basic reproduction number, which in our case is approximately 0.5. So based on this sort of relatively simple model, we can conclude that more than 50% of the US population should be effectively immunized to stop the pandemic. And here's some numbers that I put together quickly in the last minute. The current US population is around 328 million. Now, as of mid-February, around 50 million people will have been immunized. As of yesterday, it was actually close to around 48 million with either the Pfizer or the Moderna vaccines, at least the first dose. Now, in addition, more than 27 million people have already been confirmed infected through testing, and all recovered ones certainly effectively immune. So therefore, the US needs to immunize now at least 87 more million people. And with the current rate of about 1.5 million people being immunized per day, it would take about 60 days to reach that threshold. So it could be even faster, actually, given that there must be a significant fraction of the US population that is immune without being sort of officially confirmed. This is, again, a simple model, but well, we can still hope that by mid-April, in about 2 months, we may be able to kind of see the light at the end of the tunnel, and once this threshold has been reached. And finally, this project may serve as an example of solving a real-world problem, of great importance that everyone certainly can relate to. Also, an example of doing some exploratory data analysis and visualizations to explore some real data, and then using this real data to fit a mathematical model and make some predictions. This project also could serve as the basis for calibrating more sophisticated models to real COVID-19 data, and one of these models could be the SIR model. My goal is and hope is to develop this project into actually a SIMIODE model and publish the materials on SIMIODE, hopefully, including all the R code that generated all the results and the graphics, maybe through source R Markdown document, in addition to sort of having either PDF or HTML that you can produce from the source Markdown document. Maybe even Primus could be a sort of a great option to consider. And that's really it. So
well, thank you in one of those languages. We're a bit short of time, but is there any question? Brittany says, I have a clarification. Isn't the cumulative infected, I should read it. Isn't the cumulative infected from the data different from I in the model, since I in the model would be the number of infected on a given day instead of cumulative?
Well, so basically the data, the way the data is sort of accumulated, it provides only daily numbers, right? It provides only daily numbers. So you do have to accumulate these daily numbers to see the current level of infected people.
Okay, all right. And I have one question. When you're introducing the language R to students, which is free, et cetera, how do you do it? Do you just give them some sample codes and
Well, good question. Yeah, it's not easy, to be honest, but I've been doing this for many years, and I've developed lecture materials, basically a very quick sort of 20-page or 30-page now introduction to R. Also sort of an HTML with well-selected videos introducing R from DataCamp, which has a free course on R. And so I embedded all these videos in these documents. And they're 5-minute short videos, so the students can kind of watch them quickly. And I do spend a couple of lectures sort of introducing the basics of R, as well as how to work with our Markdown documents inside RStudio, because it's a great technology. This is sort of the equivalent of the Jupyter notebooks on the RStudio side, but you can also run Python code inside R Markdown in addition to R. And I've tried also with Python to mix Python and R. It works just great. Again, something similar to the Jupyter notebooks, but the RStudio Markdown technology basically has been developed to provide more control over the publication quality. And in fact, I use it even to write my papers, my presentations, everything, because it gives you a lot more control to create publication quality documents, reports, papers, even lecture notes I'm using, exams, everything.